Protein synthesis by Daniela Nutter. Protein synthesis has 3 steps. The first step is DNA and nucleic acid. DNA is a molecule that carries most of the genetic instructions. It consists of 2 bipolymer strands coiled around each other to form a double helix. Inside this double helix, we have the sugar phosphate backbone, which contains 4 out of the 5 nucleotides, which are the ones right here at the left, which are adenine, thymine, guanine, and cytosine. The 2 will always be paired up together because they're opposite, and the same goes with adenine and thymine. Then we have the monomer of DNA. This monomer contains a phosphate group, it contains a sugar, and then it contains a nitrogen base which contains any of the 4 nucleotides found in DNA, but in this case it's adenine. At the left, we have a diagram of purine and pyrimidine. Purines are the ones that have 2 rings, and they're adenine and guanine. And then pyrimidine is the one that has 1 ring, and it's thymine and cytosine. The next step in protein synthesis is the transcription that occurs in the nucleus. The most important thing in this procedure is the RNA, which is a small piece of nucleic acid that codes, decodes, controls, and expresses the genes. RNA polymerase is also known as the DNA dependent. It is an enzyme that produces primary transcripts RNA. In cells, this is necessary for constructing RNA chains using DNA genes as a template. This will allow the DNA to unzip and let the mRNA come in. The mRNA stands for message RNA. The mRNA will come in and create a complementary code that uses uracil instead of thymine. Guanine and cytosine are paired up together, thymine and adenine are paired up together, but adenine is paired up with uracil. Finally, the mRNA can go out through the nuclear pore into the cytosol and attach to a ribosome to be read. The last step is the translation that occurs in the cytosol. First, the mRNA binds to a ribosome. Ribosomes are perfect for this process because ribosomes are protein-making machines. This is the tRNA, which will transfer the right amino acid to the ribosome to build protein. The tRNA has an anticodon which matches up with every 3 nucleotide sequence located in the mRNA, which is called a codon. The pink circle is the amino acid that corresponds to the specific sequence. This is the start codon that is usually AUG. Another tRNA moves in and matches with the next mRNA codon. The amino acids will bond together and will create a polypeptide bond. The first one detaches from the mRNA so the next one can come in. This process will occur until a stop codon is reached. The polypeptide protein, which is in primary structure, is ready to be detached and to be folded into secondary structure. Protein is done and it is ready to be functional. The rest of the mRNA can leave the ribosome.